Ready?
Yes.
Go. An estimated of 26 million animals are used every year in the United States for scientific and commercial testing. However, have we ever asked animals how they feel about this? The affirmative supports the resolution that we should eliminate the use of animals in scientific research. There are 3 reasons why we believe this should stop. Number 1 is that animal testing is brutal and inhumane. There are 3 particular problems that we are going to focus on. So point A, animal testing is painful for fauna. According to the Human Society International, animals used in experiments are commonly subjected to force feeding, forced inhalation, food and water deprivation, prolonged periods of physical restraint, and the infliction of burns and other wounds to study the healing process, the infliction of pain to study its effects and remedies, and killing by carbon dioxide, asphyxiation, neck breaking, decapitation, or other means. In the animals and people's defense, the participation of humans and animals being drug tested is not appropriate. For the first point, animals being tested is an act of being cruel and inhumane. Even when alternatives to the use of animals are available, the law does not require that they be used, and often they are not. And that brings me to my sub-point B. And that is not the only problem. All producers, even those considered mild, have the potential to cause the animals physical as well as psychological distress and suffering. The HSI states that the Drake's eye test, used by cosmetic companies to evaluate irritation caused by shampoos and other products, involves rabbits being incapacitated in stocks with their eyelids held open by clips, sometimes for multiple days, so they cannot blink away the product being tested. The commonly used LD, lethal dose 50 test involves finding out which dose of chemical will kill 50% of the animal being used in the experiment. So we're not only testing on animals, but we're also practicing and getting better on our torture skills. After enduring this terrifying, painful procedure, animals are then usually dumped back into a cage without any painkillers. There is a third problem that goes along with this that is malevolent and disgusting, disturbing, which is sub-point Z. Animal tests do not reliably predict results in human beings. Yes, by testing on animals, it sometimes helps humans' disease and other illness. However, animal tests does not constantly forecast results in human beings. According to the neurologist Hasha, over 100 drugs that were effective when tested on animals have failed in humans, and over 85 HIV vaccines fail in humans after working well in non-human primates. Animals are infected with disease that they would never normally contract. Tiny mice grow tumors as large as their own bodies. Cats are purposely blinded. Rats are made to suffer seizures, and primate skulls are cut open, and electrodes are implanted in the skull. Experimenters force-feed chemicals to animals, conduct repeated surgeries on them, implant wires in their brains, crush their spine, and much more. Also, in 2013, in a study published in 2013 by the National Academy of Science of the United States of America, found that nearly 150 clinical trials of treatments to reduce inflammation in critical ill patients have been undertaken, and all of them failed. Despite being successful in animal tests, another study in 2013, a study done by Archives of Toxicology, stated that the low productivity of animal experiments in research areas allowing direct comparison of mouse versus humans data puts strong doubt on the usefulness of animals data as key technology to predict human safety. We are torturing, afflicting pain, and using animals as test models to find tentative cures that we're not even 100% sure that they will produce consistent results. And how do we know all this? Well, in contention number 2, which is my inherency claim, 95% of animals using experiments are not protected by the Federal Animal Welfare Act. And there are 2 particular problems that we're going to focus on. So point A is scientific research is done on banned animals. According to the Human Society International, The AWA does not cover rats, mice, fish, and birds, which comprise around 95% of the animals used in research. The AWA covered 11,347 animals used for testing in fiscal year 2010, which leaves around 25 million other animals that are not covered. These animals are especially vulnerable to mistreatment and abuse without protection of the AWA. Even animals that are protected under the AWA can be abused and tortured, and the law doesn't require the use of valid alternatives to animals, even if they are available. We're not only abusing these animals, we're also breaking the system that protects them. And it only gets worse, and that's point B. Suffering towards animals can also occur when given research protocol is not well justified scientifically. According to the U.S. law, in animal experimentation, animal welfare laws must address three main ways in which unnecessary animal suffering can occur in the context of medical experimentation. An experiment that was so badly designed that it could only never generate any useful scientific knowledge would never warrant animal suffering. Harder cases result when the amount of suffering is intensified down, or the experiment's potential to generate useful knowledge is increased up. Also, as a second point, they stated that unnecessary suffering can occur when the amount of animal suffering induced by an experiment is not strictly required to conduct the experiment. A legal frame seeking to avoid this kind of unnecessary suffering will encourage or require researchers to use the three R's: reduce, replace, and refine animal testing. And that brings me to my last and third argument. The use of alternative testing methods, such as the use of human cells, can replace the use of animals in scientific research to stop the slaughter of animals, which is my solvency claim. And there are a couple of alternatives that we want to focus on. So point A, the study and use of cell cultures can produce more relevant results. According to the animal replacement science, in vitro, in glass testing, such as a studying cell way of life in a petri dish, can produce more relevant results than animal testing because human cells can be used. Microdosing, the administration of doses too small to cause adverse reactions, can be used in human volunteers, whose blood is then analyzed. Artificial human skin, such as the commonly available producing epidermis, and then there is made from sheets human cells grown in the test tube for coccidials, can produce more useful resources than testing chemicals in animal skins. Thanks.